Hey VH Bio Kids, this is Mr. Wolf, and today we'll be talking about the Osmosis in Plant Cells lab. For this lab, we'll be using potatoes as our plant cell sample. To get these samples, we'll be using a cork borer. Now we have to be very careful with these cork borers because they're very sharp, and we want to make sure we only get potato in our samples and not potato in some of your skin. Once we have our cork borers, or our potato cores, we're going to be massing them using electronic balance. You'll be massing 3 cores for each sample or each solution your group will be testing. Now if you look at the picture above me, this is a picture of what potato cells look like underneath the microscope. They have been stained so that the starch vacuoles show up in purple. What we don't see is the large central vacuole that's just going to be filling up or losing water throughout this lab. You can faintly see the outline of the cell wall, which would have the cell membrane sandwiched up right close to it. Once we have our potato cores, we'll be adding them to 3 different sucrose solutions. Now we're going to be keeping the concentration of these sucrose solutions a mystery until the end of the lab. So you'll be given either A, B, C, D, E, F, or G. That's 6, right? A, B, C, D, E, F, or just through F. Your group will only be doing 3 of them. After 24 hours, we'll be taking those samples out and remassing them to find our final masses. Now during those 24 hours that your potato cells are sitting in the sucrose solutions, the cells will be losing or gaining water. If we have placed your plant cells in a hypertonic solution, we should expect that those cells will lose water, therefore losing mass. If your plant cells were placed in an isotonic solution, we should expect that the cells neither gain nor lose water at any different rate, and those cells should stay about the same in mass. Now, if we have placed your plant cells in a hypotonic solution, we should expect those cells to gain water into their large central vacuole as pictured here, and those cells should gain mass. Once we have collected all the data from this lab, we'll be calculating the percent change in mass. This is a pretty easy equation to follow, and you can see it above me right here. What you'll be doing is taking the final mass minus the initial mass divided by the initial mass times 100. You'll enter these values in the data table you'll find in your lab as pictured below me. Once you have all the percent changes in mass entered, you're then going to rank the solutions from least concentrated to most concentrated. I'll let you figure out how you're going to rank them when you do the lab. Once every group has ranked their solutions, I'll actually give you the actual sucrose concentrations of the solutions that have been tested. From this data and the class averages of all the changes in mass, you guys will be creating a graph as pictured below me. This graph will be the percent change in mass of the potato cores at different sucrose molarities. We're just going to call them concentrations. From this graph, we should be able to interpolate what the actual concentration of our potato cells actually are. This is where our best fit line crosses the 0 mark in our percent change in mass. Well, I hope you guys found this helpful and look forward to doing this lab with you tomorrow.